Okay, well, let's go ahead and get started here. So let's talk about the specific types of tissues now. We'll start with, traditionally, we tend to start with epithelial tissues, so I'll start there. Although on my review video, I actually start at the easier tissues, but we'll get epithelial out of the way. So epithelial tissues, like we said, they're often tightly connected, right? And those connections, there are some relevant concepts that you need to be aware of there. Okay, what actually anchors those connective tissues together? Okay, well, there are certain molecules. There are special molecules that can help them. Okay, there are what we call CAMs, or cell adhesion molecules. Okay, and these can basically form two general types of what we're called junctions. Okay, they can form kind of like a Velcro, if you want to look at it as a Velcro, or what we call a tight junction, where they're tightly held together. Not a whole lot of stuff is going to get through that junction. Okay, so those are your tight junctions. You also have what are called gap junctions. And gap junctions are very important because gap junctions involve some sort of channel, some sort of protein channel. Okay, so if I have a cell or two cells in a tissue that are tightly bound together, okay, so I have a tight junction, okay, there's not going to be a whole lot of diffusion or exchange of ions or particles between those cells, but some cells, okay, may have a gap junction, and that gap junction involves some sort of channel, all right, some sort of channel that allows these cells to communicate, okay? And oftentimes it's an ion channel that allows ions to move from one cell to another. In contractile tissues like muscle, that gap junction may have a sodium channel. And in fact, when we looked at muscle tissue and we looked at cardiac muscle and we saw the intercalated discs, an intercalated disc is a type of gap junction that has a sodium channel and that allows sodium ions to flow from one muscle cell to another muscle cell. This is in cardiac muscle. And you will learn why that is important a little later on. Okay, so you have these various cell adhesion molecules, and you can have these tight junctions, these gap junctions. Okay, so general types. Our general types of epithelial tissue really come down to your glandular tissues and your covering and lining tissues. Okay? So again, we're talking about epithelial tissues. Okay, epithelial. So we have glandular tissues. This should not be confused with a node. A gland is not the same thing as a node. Okay? Okay, lymph nodes are not glands. Okay? So when somebody has a swollen lymph node and they say, oh, my gland is swollen, no. Okay? So lymph nodes are not glands, glands are not lymph nodes. Okay? Like we said, glands are special structures that do what? They secrete and they excrete. Okay? So glands secrete. Okay, and they excrete. What's the difference between secreting and excreting? Seems like the same thing to me, doesn't it? The difference is when a gland secretes a substance, that is generally a substance that is helpful. Okay, it's some kind of substance that the cell wants. When a cell excretes, that is a substance that the cell doesn't really want. So excretion generally involves getting rid of some sort of waste product. Okay, when I excrete something, there's some sort of waste in it. And secretions generally do not have wastes in them. Okay, you guys okay with that? It's a difference between secretion and excretion. Okay, so like we said, what two types of glands do we have? We have endocrine and endocrine and exocrine. Okay, endocrine glands, where do they secrete?
Third space.
Hmm?
Third space.
into the interstitial space, right? And they do not have ducts. There are no ducts. Okay, no ducts, and they secrete hormones into the interstitial space. And those hormones eventually diffuse out into the blood and are transported through the circulatory system to wherever they need to go. Exocrine glands, on the other hand, have a duct, okay, and they secrete, or excrete in some cases, where? Out to a surface. All right. So you have your actual gland, you have a duct, and then that duct opens up onto a surface, and you have excretion and secretion occur. You guys okay with that? Okay, so endocrine, no ducts, hormones, interstitial space. Exocrine have ducts, okay, and they secrete out onto a surface. That may be out onto the surface of your skin, it may be out onto the surface of your respiratory tract, maybe onto the surface of your gastrointestinal tract. You guys okay with that? It may be the surface of your urethra. Okay, that's the basically the inner lining of the tube that urine comes out of your body in, or the vagina as well, right? You may have secretions there as well, and glands there as well. Okay, cool. And then you have, what's the other genotype? So you have glandular epithelial tissue, and you have your covering and lining epithelial tissue. And how do we identify covering and lining epithelial tissue? What are the two general categories that we use, or criteria that we use to identify specific types of covering and lining epithelial tissue? You're on the right track. You identified a type, but how did you identify a type? Okay, so layering is one of the two. And then what's the other two? Layers and shapes. Okay, layers and shapes. Okay, and I'm just going to talk about shapes first. You have a couple of different shapes, or three rather. You have three general shapes. Okay, you have a shape that is flat and thin, and has a flat, thin nucleus, and that is known as squamous. So squamous epithelial tissues have flat, thin cells. Okay, and then you have a shape that is about as high as it is long, and that is cuboidal. Okay, so you have cuboidal epithelial tissue. And you also have cells that are taller than they are wide, and these are known as columnar. All right? So I've got squamous, cuboidal, and columnar. Okay? And then what are the general types of layering? You have what's just one layer of cells? Simple. That's what we call simple, right? Simple epithelial tissue is one layer, and then if you have multiple layers, we call that stratified. Okay. So you can have simple squamous epithelial tissue. You can have stratified squamous. You could have simple cuboidal. You could have stratified cuboidal. You could have simple columnar. And you can have stratified columnar. You guys cool with that? Okay. Now, there are some special cases, though, that we need to be aware of. There are two special cases. The first special case is a type of stratified epithelial tissue where it has all different shapes. These are the cells that can take on multiple different shapes. And why can they take on these different shapes? Because they are stretchy. They can stretch. And their stretchiness means that their shape can change substantially. Okay? And this is a type of epithelial tissue. Again, this does not fall into our general categories. Okay? This is known as what? Transitional. There you go. Transitional. Transitional epithelial tissue. The other odd man out, so to speak, odd cell, odd tissue out, is something known as pseudo-stratified columnar. And what we see there, the nuclei are not lined up, but rather the nuclei are at different locations within those columnar cells, and it gives the appearance that they're stratified when they're really not. And that's what pseudo means. Pseudo means false. So these are falsely stratified, okay? But pretty much all the other types of epithelial tissue, we use layers and shapes, okay? You start with the layering, and then you finish with the shape. So you would call it simple squamous. You guys okay with that? So far, so good? Okay, you're okay with the surfacing? You have the apical surface, you have your basal or your basolateral surface, as some people may say, and that is attached to a what? A basement membrane. Generally, that basement membrane consists of connective tissue. Okay, so, and these tend to be, in general, these tend to be avascular, right? Avascular, tightly packed. Okay, so let's talk about some examples. So the first example that I want to talk about is simple squamous. Simple squamous epithelial tissue. So it is one layer of squamous cells. Okay? Where do we classically find simple squamous epithelial tissue? The lungs. Specifically the little air sacs called the alveoli, right? Why would we need to have simple squamous epithelial tissue in the alveoli? Diffusion, there you go. Gas diffusion, gas exchange. Right? Does that make sense? Again, form following function, right? Okay. Thin, unstratified layers. Okay, that's the form, and the function is, it facilitates diffusion of gases in and out of the body. Okay. There is also another area where we see simple squamous, or is another area where diffusion is important. Certain blood vessels, right, like the capillaries. They're really tiny capillary beds where diffusion in and out of the blood occurs. So the capillary beds. All right. So the alveoli and the capillary beds. All right, you guys cool with that? So we'll just stick with simple. Okay, so let's talk about the next one. Simple cuboidal. Okay, so this is one layer of cuboidal tissue cells. Where do you think we would see this? So, say again? Yeah, kidneys. So we have diffusion here. Okay, so you have diffusion here. It's really important. But whenever you have excretion, okay, you're trying to get rid of stuff, cuboidal cells are important. And so simple cuboidal epithelial tissue tends to be in the tubules of the kidneys. Okay, that's a very classic, because we have excretion occurring there in the tubules of the kidneys. You guys cool with that? So far, so good? All right, well, let's just go with the last of the simple tissue, and that is what? That is simple columnar. Simple columnar epithelial tissue. Where do we tend to find this? GI tract, yeah, the GI tract. Specifically, the intestines. Okay, this is a type of tissue that lines the inner surface of our intestines. And when you look at the intestines, what you see, the gross anatomy, if I have my intestine here, all right, and I cut a section and I open it up, okay, what you're going to see is you're going to see something kind of like this. Okay, so imagine that it's coming out of the board at you there. So I've taken a cross section of it. Okay, you see these little finger-like extensions. These are called villi, or villus if you're talking about one. And then what happens is when we zoom into these, okay, when we look at just one of the villi, one of the villus, they actually have a microscopic structure, a microscopic little finger-like structure called microvilli. And the microvilli themselves are covered in this simple columnar epithelial tissue. You guys cool with that? Does that kind of make sense there? Okay. And yeah, this is classic where we see this. So what's the big thing here? Absorb nutrients. Okay. Absorbing nutrients, the big thing that we see occurring with simple columnar. So far so good, guys? All right, so let's now talk about stratified, and we will move to stratified squamous. And where do we classically find stratified squamous epithelial tissue? In the what? Huh? Your skin. There you go. Yeah, your skin. The outer part of your skin, right? No, also known as the epidermis. And this is actually what we'll be talking about lab this week. Skin, epidermis. Now you have two general types of stratified squamous epithelia. You have types that have keratin in them, right? And we call that keratinized. And non-keratinized. Keratin is a hard protein, right? It's a hard protein that resists mechanical stress. Okay? So where do you think you would have your keratinized stratified squamous? Where at? Your skin itself, right? Okay. Where would you expect to see non-keratinized? Okay, where you don't have is much of these mechanical stresses. So in your mouth, right? You generally don't walk, you generally don't run a marathon with your mouth, right? So you don't have that mechanical stress. Good. Where else? Your mouth. Yes, your esophagus. Okay, good. Your esophagus can have this. Some smooth muscle. Good. Where else do I have skin? What about the anus? Right? The opening. And your vagina as well, right? You have skin there. But in general, you don't have significant mechanical stress there. So that skin is going to have, is not going to be keratinized like the skin that you have, like your skin, if you will, your skin proper. You guys cool with that? Mm-hmm. All right, cool. I will briefly mention stratified cuboidal. This is a fairly special case. We only see it in a few specific couple specific areas of the body. So stratified cuboidal is multiple layers of cube-like cells, right? Where would we see this? One of the areas that we see this is actually in the tubules of the testes. And some glands, specifically the sweat gland. Nice pickup there. Yes, sweat glands. Okay. Other than that, you do not tend to see stratified cuboidal epithelial tissue. All right. Let's talk about our special cases now. Let's talk about our transitional. Where do you suppose we see a lot of transitional epithelial tissue? Needs to be lots of stretchiness going on. Say it again. There, the urinary bladder, right? Because that bladder stretch out, to the size of a grapefruit or bigger, and then it shrinks to about the size of a walnut, right? So you need to have specialized epithelial tissue that's really good at changing its shape. So the urinary bladder. The urinary bladder, yeah. Okay, and then finally, we'll talk about the last one, and that is the pseudo-stratified columnar epithelial tissue. This is classically found where? Where? In the airways. Yeah. In the airways. Not the alveoli, that's your simple squamous, but the airways, the passages to the alveoli, are lined in this pseudostratified columnar epithelial tissue. Okay. And the pseudostratified columnar epithelial tissue tends to be covered in what? Cilia. Tend to be covered in cilia. And then on top of the cilia, okay, so here I have cilia here. And then I have specialized types of cells that kind of, I imagine they kind of look like a wine glass when you look at them. You guys are familiar with a wine glass, right? They kind of look like a wine glass. And what's another name for a wine glass? Old school name. A goblet. And these are known as goblet cells because they kind of look like wine glasses. Okay? And these goblet cells are specialized pseudostratified columnar cells that do what? They secrete mucus. Okay, they secrete mucus, and that creates a blanket of mucus that sits on the apical surface, okay, of this tissue. And then what happens is the mucus is very sticky, right? And dust and disease, foreign particles, viruses, bacteria can stick to this, get trapped on this mucus, and then the cilia beat like this, and they push all that mucus up to the upper airway with all that nasty stuff attached to it, and you either swallow it or cough it up. We call expectorated. And that's known as the mucociliary escalator, or the mucociliary transport mechanism. Okay? Very important part of your airways. This is what keeps our lungs clean, keeps us from getting pneumonia in most cases, and when this fails, and it backs up, our risk for infection in the lungs increases quite a bit. And anybody who smokes, of course, exposure to cigarette smoke in particular can disrupt the mucociliary escalator and prevent the cilia from moving that mucus blanket up. And you guys are all familiar with the quote-unquote smoker's cough. You ever been around somebody who smokes, and when they get up in the morning, what do? They hack and hack and hack, and they start coughing up these big globs of mucus that basically can't get cleared from the lungs, right? So yeah, there you go. You guys go okay with that? So far so good? All right, cool. Well, let's move right on. The next type of histology is pretty simple. I think you guys will be okay here. And this is not epithelial tissue, but this is muscular tissue. All right, and I'll just write it here. So this is all epithelial. This muscle tissue is easy compared to epithelial tissue. All right, so muscle tissue. You have what? How many types of muscle? You have 3 types, right? And muscle tissue, as we said, is highly vascular, as opposed to the epithelial tissue, which is generally avascular. Okay, so muscle tissue, you have skeletal, cardiac, and smooth. All right. And skeletal muscle tends to have what kind of structure? A long, thin fiber-like structure, and it is striated, right? So long, thin, striated. All right, these are sometimes known as muscle fibers. All right, so long, thin, striated. The striations, of course, are caused by the actin and myosin fibers in a particular arrangement. Cardiac muscles, oh, and skeletal muscle, how many, is it multi or uninucleated? Multinucleated. Yes. Multinucleated. Cool. All right, cardiac. Cardiac muscle is different from skeletal muscle in that it is branched, right? It is branched. It has those intercalated discs. Is it also striated? Yes. Yes. Okay, so both skeletal and cardiac muscle are striated. So you cannot use the presence of striation to differentiate between these two. But if you see striation pattern, then at least you can narrow it down to skeletal versus cardiac. And then you go, okay, is it branched? Does it have intercalated discs? Yes, yes. Oh, it is cardiac muscle. All right. And then smooth muscle is kind of the odd one out. These are short, kind of spindle shape. They're short, they're spindle shaped, they do not have significant striation, so they lack striation patterns. Okay, so they kind of have a short, spindly kind of appearance, uninucleated. We got to look at that under the microscope. Okay, you guys okay with that? Is that good to go? That's muscle histology, really, really easy stuff. All right, tell you what, you okay if I erase the muscle? Okay, let's talk about the next easiest one, and that is nervous. Okay, again, it's easy to talk about, and then I will save connective tissues for last. How does that sound? We'll save the worst for last, so to speak. All right. So muscle and nervous tissues, fairly easy to talk about. Connective tissues and epithelial tissues are a bit more nuanced. Okay, so let's talk about nervous histology. Okay, what 2 types of cells do we have? You have neurons and glial cells. I'm going to talk about the neurons in just a little more detail. Okay, the basic structure of the neuron has 3 basic components. Okay, it has that kind of that star-shaped, that larger star-shaped component to it. Okay, and that's known as the body of the cell. Another term for body, and it is a term that is commonly used, is soma. Soma means body, okay? So you will see those terms used interchangeably. You have the cell body. This contains the nucleus and the other organelles of the neuron. Then there are generally a series of projections from the cell body that come out of the cell body. All right. And these projections are responsible for bringing information into the soma. Okay, so information comes into the neuron through these projections, and these projections are known as dendrites. All right, dendrites. Dendrites bring the information into the cell body. Processing of that information occurs in the cell body. And in neurology, we'll talk about that process in a lot more gory detail. And then information leaves the neuron generally through a long projection known as an axon. Okay, so information comes in through the dendrites, and information leaves from the axon. Okay. That's all the detail we need to know about the neuron at this point. Okay, but we do need to know a little bit about the glial cells. Okay, there are two general types of glial cells. You have what's known as your macroglia. So these are tend to be larger, and your microglia, or glial cells. Okay? There's really only one type of microglia, or microglia, and that's what we call microglia. Okay? And these are actually, believe it or not, these are immune cells. These are immune cells. They are specialized, what we call macrophage. The microglia are actually a type of macrophage. How confusing is that, right? A macrophage is a type of white blood cell known as a monocyte. And there is this wasn't a really good movie, but it was kind of funny. It was a cartoon. I always say it's Chris Rock, but I don't think it's Chris Rock that does the voice. But it's Bill Murray. And yeah, Bill Murray is it's called Osmosis Jones. Okay, Jones is a cop, and Bill Murray is the guy. He's a zookeeper, right? And the egg, he's going to eat an egg, and it falls into this pit of monkey feces, and the monkeys are laughing at him. He grabs the egg out of there and eats it. And he's like, "Ha, I got the last word." And anyway, there's a virus on that egg. Anyway, Osmosis Jones he's a cop, right? And what's his superpower? He can kind of pop in and out of all these other tissues, right, and morph his body around. And he's actually a monocyte. And when monocytes leave the blood and go into the tissues, they mature and become what we call macrophage. So macrophage are matured or monocytes that have left the blood. So the microglial cells are these immune cells, or macrophage. Okay, and then it's the macroglia that I'm going to talk about, and there are several types of macroglia. And let me just choose a different color to do that in. Maybe a nice blue will work. Okay, there are 4 types of macroglia that I want you to be familiar with. Okay, the first type are what are known as astrocytes. Astrocytes. Okay, astrocytes are the most abundant glial cell in the body. Here, get down here so I can continue writing. Okay. These are very important when it comes to regulating the external environment around the neuron. Okay, they make it a bit friendlier for the neuron to live in. And they also, we think they may make up part of a special structure known as the blood-brain barrier, which is something we'll talk about in more detail in neurology. Okay? So those are your astrocytes. Okay? You also have what are called oligodendrocytes. Okay, your oligodendrocytes. Your oligodendrocytes exist in very specific areas. Okay? And oligodendrocytes make a substance that is very important for neurons. It's a fatty substance called myelin. And they make myelin there we go, myelin. And the myelin wraps around the axon and it creates what's known as a myelin sheath. Okay? And that is a kind of insulator. It insulates the axon and allows this signal to travel through the axon more efficiently. Okay? In the central nervous system, the CNS, that's the brain and spinal cord, oligodendrocytes make myelin. Now, we have nervous tissue outside of our central nervous system, right? Do you have nerves in your arms and your legs and your hands and your feet? Those nerves outside your brain and spinal cord, that's what we call the peripheral nervous system. There is a different type of glial cell that makes the myelin for those nerves, okay? And that is called a Schwann cell. A Schwann cell. And Schwann cells do exactly the same thing as oligodendrocytes. They make the myelin, but they do it in the peripheral nervous system for nerves outside of the brain and spinal cord. Okay? And then the fourth cell type that you just want to be familiar with is something called the ependymal. Or ependymal cell. And this is located in a specific area as well. It's not throughout the entire, it's located in very specific areas. And these cells make a kind of fluid called cerebral spinal fluid, or CSF. And this cerebral spinal fluid is a fluid that surrounds the brain and the spinal cord. And so your brain, your spinal cord kind of floats in this fluid that kind of protects it. And it can kind of move around a little bit, and there's some protection that occurs there. And that cerebral spinal fluid is produced by these ependymal cells. Okay? You guys okay with that? Now, when you look at a tissue slide of nervous tissue, you will not be required to differentiate astrocytes and oligodendrocytes and ependymal cells. Okay? When you just look at a slide of nervous tissue, all you can really see are the neurons and the glial cells. And remember looking at that in the lab. Okay? So far, so good? Okay, take a break. Take about, let's see, 7 minutes, and then I'll see you guys back. We'll start on connective tissue. I don't know if I'll be able to get all the way through connective tissue because there's a lot to talk about there.